Hello, and welcome to our presentation on SARS-CoV-2 and its unique pathogenetic properties. It's presented by me, Aaron Satchwell, and George Desmestihas.
Now, what we'll be discussing is the viral origins of SARS-CoV-2, its means of initial infection and what that means, the level of pathogenicity that it expresses, and its drug resistance, and kind of what makes it able to resist drugs and resist treatment as it does. Now, when a virus such as SARS-CoV-2 starts a world pandemic, many people begin to wonder, where did this come from and how did this happen? In the scientific community, it's a general consensus that the coronavirus was able to infect humans via zoonotic transmission. Now, what zoonotic transmission is when a virus such as SARS-CoV-2 jumps from an animal species into a human. Thus infecting them and allowing the virus to infect the human population. Now, this may have happened by the virus infecting an animal such as a bat, and that bat being consumed through food. The virus is theorized to have started in Wuhan, China, and that's where infections of humans began. And there's oftentimes wet markets where animals such as bats or pangolins are sold and people eat them for food. Another way that SARS-CoV-2 may have infected humans is just direct contact. So perhaps that the virus was in an animal that humans come into contact with often, and transmission occurred that way. Another theory put forth is that the virus jumped into an animal such as a bat and then into another host and then into the human host. Research that supports this theory of zoonotic transmission is the fact that in the wild, animals have a wide variety of coronaviruses that they can also be infected with, and it has been found that the nucleotide similarity in those coronaviruses is extremely similar to that of the coronavirus that infects humans. Furthermore, it's been found that the receptor binding domains, which is the region through which these coronaviruses can bind to their hosts, are extremely similar in the animal coronavirus variants as they are in the human one.
Initial infection of the SARS-CoV-2 virus is transported via respiratory droplets through either a sneeze and or cough, and this causes the aerosolization of these respiratory droplets. And once aerosolized, these particles will be transported into the upper and eventually lower respiratory tract, which will attach to the lung epithelial cells to create, which will then give rise to physical symptoms. Coronavirus's means of infection are similar to that of SARS-CoV-1, or its predecessor, in that it affects the upper respiratory tract, but the main difference between SARS-CoV-1 and SARS-CoV-2 is its ability to travel lower into the respiratory tract, allowing it to connect and infect lung epithelial cells themselves. Once infected, these symptoms physical symptoms will arise, and they include fever, diarrhea, fatigue, sore throat, as well as a wide variety of other cold-like symptoms. And in the worst case scenario, due to may result from pre-existing conditions, could result in death. Now, just a brief overview of the diagram here to the right. Again, we have the respiratory droplets, which are results of a sneeze and or cough. These particles then become aerosolized, which will then go to infect a larger population of people. Level of pathogenicity for the virus. In order to understand the level presented here, you must dive first into the anatomy of the virus itself. Taking a look here to the right upper hand corner, we have the virus itself, and the most important means of infection for the virus itself is known as spike proteins. These are often abbreviated as S proteins, but more specifically known as spike proteins. And before we get into the mechanism, this S protein itself is divided into two subunits as seen below. Very briefly, we have the S1 subunit and the S2 subunit. And those make up the spike protein. Initially, this spike protein remains inactive until the virus itself, through the spike proteins in the S sub 1 unit, the S1 subunit, binds to the membrane of the host cell. As demonstrated below here, we see the S sub 1 unit binding to the membrane of the host cell. And through the use of two enzymes, you have TMPRSS2 and the furin enzyme, which work together in a cleavage process to cleave the S1 from the S2 subunit, which will release the S1 unit away, leaving the S2 subunit to proceed in the viral capsid fusion with the membrane of the host cell. And next, we have the actual means of infection for this virus. When we take a closer look at the S protein on the tip of the S1 subunit, we have what's known, we have a receptor binding domain, which recognizes a receptor known as the angiotensin converting enzyme receptor 2, also known as ACE2. This receptor is present in the intestines, it's present in the heart, the kidney, and most importantly for this virus, the lungs. And this virus will bind to this receptor with very high affinity, which demonstrates its level of pathogenicity, and it's due to its effective level of infection, successful infection with the high binding affinity to these receptors. And with this receptor, the coronavirus is allowed to, or with this high affinity level, the virus itself is allowed to descend lower into the respiratory tract, again, infecting the lung epithelial cells. And it's compared to its predecessor of SARS-CoV-1, it proceeds much lower, infecting those lung epithelial cells.
About the SARS-CoV-2 virus is the fact that it's seemingly drug resistant. Many medications and therapies have been used in an attempt to reduce the symptoms and the spread of SARS-CoV-2. However, nothing as of yet seems to be very effective. There are many reasons as to why this happens. The first being that the virus is novel, so we haven't yet developed proper treatment for it since it's so new and spreads so fast. Another reason is the fact that SARS-CoV-2 is an RNA virus, which means it'll have a very high mutation rate. And another reason is the widespread use of biocides and antibiotics, which we'll get into later. One aspect of SARS-CoV-2 that makes it unique is the fact that it's an RNA virus. So an RNA virus is a virus whose genetic material is stored solely in the form of RNA. Additionally, RNA viruses are able to mutate more rapidly because they have no DNA in them, and thus they forego the transcription process when they're replicating. Additionally to that, RNA viruses have no proofreading mechanism typically, which means that if a mutation in a sequence occurs, there's no proofreading mechanism to correct that mutation. As a result, mutations will move on to the next generation. And because of natural selection, only those mutations that are beneficial to the organism will be passed on to the next generation. Essentially what this means is that an RNA virus, such as the viruses in the coronavirus family, are able to readily adapt and mutate in order to live and survive in their environment and in fact thrive. And this is what happened with the SARS-CoV-2 virus. It was able to undergo a series of smaller mutations, making it go from a non-virulent coronavirus strain to a virulent SARS-CoV-2 strain that is infecting humans today. Now, what this means for drug resistance is that when introduced to a new environment, such as the environment that a drug can create, in the human body, for example, SARS-CoV-2, because it mutates so rapidly, is able to undergo changes that allow it to resist that environment and survive in it still. Now, what this means is that there's potential that even if a vaccine is found for SARS-CoV-2, that it may undergo more mutations, allowing it to resist the vaccine and continue its spread among the human population. Now, one of the reasons that SARS-CoV-2 seems to be very resistant to medication and treatment is the fact that the widespread use of biocides, antibiotics, and other medications to combat SARS-CoV-2 may set it up to select for resistance to such medications. Essentially, what this is saying is as SARS-CoV-2 is exposed to more medicines and antibiotics in an effort to treat it, it may actually develop resistances that'll help it to treat it other medications in the future. So how this occurs is when a virus such as SARS-CoV-2 is exposed to antibiotics or antivirals or drugs, such as those that are used in humans or those used in animals and pets, as well as being exposed to such things from pollution in the environment, it'll eventually begin to develop and mutate, especially since it's an RNA virus, the ability to withstand those chemicals and medications and actually thrive in those environments. In part, this is also because of globalization and increased imports and exports as well as travel. Since many people, many animals, and as a result, many pathogens, drugs and treatments are able to cross international borders more readily. So here we have this table that shows how does resistance spread, and it goes from antibiotics, for example, used on humans to those used in animals such as livestock and pets. And then we have pollution from a medicine producing factory, for example, and then globalization, imports and exports between countries, and finally travel. Despite the fact that SARS-CoV-2 is seemingly resistant to most medical treatments, one new treatment has been developed, remdesivir, which my group mate Aaron will talk about.
What is the current standard for treatment and preventative measures for this virus? First off, we have a drug that is just been approved by the FDA as of October, actually, and is known as remdesivir. And this is the first drug that is approved, again, by the FDA to treat COVID-19. And this drug is specifically a nucleoside analog inhibitor, also known as an NAI, that is responsible for inhibiting the SARS-CoV-2's RNA polymerase. To put this into perspective very quickly, this is the same process that is used in fighting against HIV and other viruses as a means of inhibition. So it's a widely used process, the NAIs. Again, CDC guidance has designated management and has made preventative measures in order to have a lower chance of getting the virus. The diagram to the right here, we see hand washing, very important. Again, washing 20 seconds allows for the soap to dissolve the bilayer as seen in the anatomy above, the bilayer of the virus itself, and face mask wearing, again, helps prevent the spread of the respiratory droplets, especially out in a public setting. Lastly is the 6 feet rule that is also important in preventing the transmission of these aerosolized molecules.
Overall, SARS-CoV-2 is a unique virus that has many interesting qualities, one of which being its zoonotic origins and how it was transmitted from animals to humans, another of which being its means of initial infection using its S protein to infect human epithelial cells in the lower respiratory tract. In addition to that, it has varying levels of pathogenicity where one person may have no symptoms and another person may experience SARS-CoV-2 interacting with their pre-existing medical condition and proving to be extremely dangerous and potentially fatal. In addition to that, it's seemingly drug resistant. Although many doctors and scientists have tried to find out medications that will combat and slow the spread of COVID, few have proven to be very effective, if any at all. Because of these reasons and more, SARS-CoV-2 is a unique virus worth being studied and will be remembered for generations to come. So here we have our resources, and these are what we used to make this presentation possible. My partner Aaron and I would like to thank you for your time in listening to this presentation, and we hope you enjoyed it.